Good evening, everyone. I'm Andrea Bodner, the Donald G. Combs Science Director at the Gloucester Marine Genomics Institute, and it's my pleasure to welcome you to the first episode of our 2023-2024 GMGI Science Hour. I'm so glad that you can join us this evening. And tonight, I'm really happy to welcome Dr. Ian Hewson from Cornell University, who will be discussing his work on marine diseases and the impact of mass mortality events, like the recent die-off of sea urchins in the Caribbean. But before the presentation begins, I want to provide some background information about our institution, GMGI, for those of you who may be new to us. And I'm going to do that tonight by sharing a video with you all. The oceans are facing tremendous challenges, from climate change to plastic pollution. We're seeing tremendous change even in our own backyard.
GMGI is a nonprofit that addresses critical challenges facing human health, the oceans, and the environment through innovative scientific research and education.
We pursue three distinct but complementary strategies that braid brilliantly together. The first is our research. We conduct marine research powered by genomics. Our education initiative is providing a career launchpad for local high school graduates who are struggling to find opportunity. And the third is our science community initiative, where we are working to create conditions to attract biotechnology and life sciences companies to Cape Ann.
With the culture and economy of Gloucester deeply rooted in maritime history, it's a perfect location.
We have a remarkable amount of talent, capital, and innovation happening all at once. Companies that have come here have realized great support from the state, the city. They've witnessed a robust, capital-efficient industrial infrastructure. Gloucester is becoming a very popular place to do business.
Our science strategy is based on a platform of advanced molecular biology and genomic technologies to address questions related to oceans and human health with three focus areas: biomedicine and biotechnology, ecosystem function and health, and fisheries and aquaculture.
What's really exciting about GMGI is the way the technology has progressed is we're on the cutting edge, and that will allow us to answer these really complex and interesting questions.
GMGI is a really incredible place to work. Not only do we have this state-of-the-art research facility right on the water, we have a really incredible team of people who have a broad range of backgrounds and knowledge.
The Gloucester Biotechnology Academy aims to democratize access to the life sciences and to train people to fill the important critical roles as professional laboratory technicians.
Before the academy, I was kind of lost in my own path. I had always enjoyed science in school growing up, but it was just something that I always felt was out of reach. The academy and the education I received has really given me a second chance. My time at the academy was spent a lot in the lab doing hands-on training, and that had really prepared me to get out in the workforce. Now I work for a company called Elevate Bio in Waltham, Mass. I am an associate scientist there in their next generation sequencing core.
They come out of this with very specific skills, practical skills, intuitive skills that are very important.
It uncovers their love of science, their passion for science. You can see the change in their level of confidence.
Coming out of the academy, I have more hands-on experience than a lot of my colleagues that did 4 years in college. My internship then led me to a full-time job for a whole year at Dana-Farber. And then after my internship and my year there, I actually joined a private biotech company where I've grown tremendously. It's a small commitment that unlocks life-changing opportunities.
We've graduated 80% of the students who we've enrolled over the past 6 years. 90% of our graduates are working in life sciences or have enrolled in college to study science.
To be able to take young people who have had very little STEM education and be able to give them a huge opportunity for career growth, to me, is very amazing and special.
We're changing the trajectory of the community, both through bringing science to the community, making new discoveries that will have an impact on fisheries, on human health, and also changing the trajectory of the students we train at the academy.
We are small but mighty. You would never know that we are a team of under 30 people when you see the groundbreaking research being conducted in our labs and the transformative education experiences being provided at our academy. And we are just getting started.
If you'd like to learn more about GMGI, please feel free to reach out to us anytime. Now to introduce tonight's speaker. Ian Hewson is a biological oceanographer, a marine ecologist, and a professor in the College of Agriculture and Life Sciences at Cornell University in Ithaca, New York. He grew up in Washington, D.C. and Canberra, Australia, and completed his undergraduate degree in marine science at the University of Queensland in marine phycology. That's the study of marine algae. He then moved to the US to pursue a PhD working on aquatic viruses and bacteria at the University of Southern California, which he completed in 2005. And after a postdoc at the University of California, Santa Cruz, Ian joined the faculty at Cornell University in 2009. At Cornell, he leads the Marine Mass Mortality Lab, which seeks to understand the causes and drivers of invertebrate mass mortality events. So please join me in welcoming Ian to the Science Hour this evening. And if you have any questions throughout his presentation, please use the Q&A function on your screen, and we'll get to those questions at the end of the presentation. So take it away, Ian.
Great. Thank you so much for that introduction, Andrea. And also thank you to everybody at GMGI for inviting me along to speak this evening. Very, very exciting to share with you some of the research results that we've had over the last couple of years working on mass mortalities, unexpected mass mortalities that appear out of nowhere. And it's up to us to basically try and figure out what's going on. Let me just share my screen to begin. Let's see if I can do this correctly. After a few years, you'd think I'd know how to do this. All right. So yes, I am indeed at Cornell University. I am in the Department of Microbiology. Please don't let that scare anybody. All right. I am a micro person, but I do a lot of marine ecology as well. And we're based in Ithaca, New York, which is on the traditional homelands of the Gayogono people of the Haudenosaunee Confederacy that precede the establishment of the United States. And that's where we actually perform our work. So tonight, what I'm going to be doing is describing some of the research that we've done, particularly focused upon this animal here, Diadema antillarum, the long-spined or black sea urchin in various descriptions across the Caribbean, and a very large sort of mortality that happened in the last year and a half. I'll start off just by sort of emphasizing why we are interested in echinoderms in general. Echinoderms are, the name comes from spiny skinned organisms. They include things like sea stars, sea cucumbers, brittle stars, and of course, sea urchins. And, you know, they're out there, they're pretty much everywhere across the entire ocean. And they do play very important roles, both in the environment as well as in our own lives. They are crucially important to many ecosystems because they are either grazers of algae, and basically they allow sort of other things to persist that algae would otherwise outcompete. And I'll give you an example of that on the next slide. But they're also economically important. There's a very large fishery for both sea urchins, which are known as uni in some cuisines, as well as in sea cucumbers in certain other cuisines. So here, for example, is a board over a highway in Shandong province in China, which basically tells people or advertising their sea cucumber products. And it's a very, very large fishery over there. So they are definitely consumed. An interesting thing about echinoderms is they have a very unique anatomy and biology. Whereas we have things like blood flowing through our veins, echinoderms, on the other hand, have seawater flowing through their bodies. All right, so they bring water in from the outside and then essentially filter it very lightly before it circulates throughout the animal. And that's what they use to transport materials around. So very, very unique. And another really cool thing is that echinoderms in general, they have very, very unique microbiomes. And we mean by microbiomes all of the microorganisms associated with an organism. So we have a microbiome in our gut as well as in our skin. These organisms, they're basically just covered in bacteria and archaea. And those types of bacteria in echinoderms are very different from most of the other invertebrates in the ocean. But perhaps the most exciting thing and the most sort of interesting thing from my perspective as to why we're studying them is that they experience these mass mortalities, which basically can lead to fundamental changes in ecosystem structure. And this is known mainly because they affect things that are basically keystone predators, all right? So this is Diadema antillarum. You can see it sort of hiding out underneath this little coral outcrop. This is a massive coral that's called Siderastria siderea. I think it's called the scarlet coral, little scarlet coral or whatever it is. And this photograph actually really nicely demonstrates its action in the environment, okay? So you have corals here, which basically compete with this stuff, which is on the substrate around it, which is algae. And if left unchecked without anything keeping that algae at bay, this coral would basically die, be covered over by the algae relatively quickly. However, having sea urchins nearby or in the environment in general, these actually consume this algae and basically allow this coral to persist, all right? And so basically what I'm saying is that diadema keeps coral reefs very healthy. And when you take these diadema away through processes like mass mortality, it can lead to big ecosystem shifts and deleterious impacts. And we know this because in the early 1980s, in about 1983, there was a monumentally huge catastrophic die-off of this particular species that used to be really abundant around the entire Caribbean region and wiped out over 99% of it, okay? It is a disease which first appeared here on the Caribbean side of the Panama Canal. It then moved more or less with currents around the basin, so up this side into the Caribbean, the Mexican Caribbean, and then across the top of South America there into the Windward Islands, and then finally up to the Florida Keys and beyond, even to Bermuda. We still do not know what caused this mass mortality, all right? In the 1980s, there weren't quite the techniques available, the resources available to investigate what was going on. And unfortunately, all of the specimens that were taken of diseased animals at the time were lost in hurricanes, or they were simply lost through trying to work out what was going on and they sort of ran out of material, okay? So we never knew what caused this, but one thing we can say is that the ecosystem changed from mostly a coral-dominated ecosystem to largely an algal-dominated system as a consequence of removing this really, really important herbivore. And so fast forward to the next 30-something years, and essentially we were sort of watching and waiting, and there was some recovery of this Diadema antillarum over that period to about 5 to 10% of what it was before this 1983 mortality. And unfortunately, in April of last year, it started again, or at least it was thought to be April. We tracked it back through citizen science records to be back in January. Started here in St. Thomas, which is in the Virgin Islands. Within sort of a couple of months, it had spread all across the Windward Islands. It also had spread towards the Dominican Republic. We don't know about Cuba, but we know that it finally reached up into the Florida Keys in about June of last year. And then eventually it made its way into Aruba and Bonaire and Curacao. Okay, so a very different pattern of distribution from the early 1980s. But the threat that it had for coral reefs really attracted our attention. We were really, really keen to work out what was going on. All right, and so that's why we became involved in this. What did it actually look like? Well, the first thing that you have to do is to basically find collaborators, find people who are sort of able to do in situ monitoring and to basically work and collaborate with these people to be able to gain samples to analyze. This project that I'm going to present today represents a huge collaboration with a lot of different moving parts and a lot of different organizations. You can see all of their logos here. Every institution and every Caribbean nation that we worked with was involved. And so lots and lots of collaborators, and we wouldn't be where we are today without being able to collaborate with scientists in the area. It also covered 11 different jurisdictions, and this includes, places like Puerto Rico, the Virgin Islands, Aruba, Turks and Caicos Islands, etc. And we're currently investigating it with a further 7 jurisdictions, countries in the area. So huge international collaboration, lots and lots of international partners involved, and super exciting to bring together so many scientists from across the region to be able to investigate this phenomenon. In terms of what it looked like when we finally sort of got down into the field, this is actually a photograph I took in April of 2022 off of St. John. these things basically should look somewhat like this, okay? Normally, they're sort of hiding out in crevices. You can see a couple up here on the top right. They're sort of hiding out during the day underneath rocks and within little caverns within those rocks. However, a large number of them at the affected sites were sort of wandering out into areas where they would be otherwise spotted by predators and perhaps, eaten. They fell over, all right? So this is not a normal orientation for the animal. This is actually on its side. A lot of their tube feet on the bottom, which the ways by which they move around, were missing. And essentially, it wasn't long until they became a pile of ossicles or a pile of spines and little plates on the bottom, okay? The timeline from when they first started to show this condition, so they first started to have unusual drooping spines and were behaving weirdly, to the time that they died was very rapid, on the scale of hours to only sort of a day or two, okay? And so this was very alarming because it meant that either there was something really, really wrong with the water, in terms of physicochemical conditions, or it was some sort of pathogen, okay? It was some sort of disease. Once these animals lost their spines, particularly on their top surface, any and all fish and other invertebrates came along, because that's their main way they defend against predation. Any and all invertebrates would come along and eat them, okay? And most importantly, when the sites were affected, over 90% of specimens they died, okay? So it was really catastrophic. There are sites out there that previously had some density of diadema that are completely gone till this day, all right? So hugely catastrophic for those areas. And so, faced with this, we worked with the Atlantic and Gulf Rapid Reef Assessment Network, which is a network of scientists that I mentioned before, to come up with some very sort of important questions. And the first of them is basically, what causes it? All right, that's the $64 million question with any marine mass mortality. What's the cause, and can we do anything about it? And so it could be anything out there. It could be, as I mentioned before, environmental stressors, changes in water quality. It could be the introduction of pathogens. Could be a new toxicant that's coming in, a new product which is being used on land that's coming down in runoff and basically is very, very harmful to these urchins. So we really didn't know going into this what was actually causing it. And then you have a couple of secondary questions, things like, why now? Why is it happening at this very moment? Has something changed very recently? Is it an invasive species? Is it something which is being introduced into the ecosystem? And then sort of as a tertiary question, it's what can we do about it? All right. How can we tell managers to come up with best practices to stop this thing from happening again, or perhaps spreading to new areas? And so my research effort is mainly on this first question, what's actually causing it? However, we have these secondary questions which are under investigation at the moment. So the first thing that we needed was to work with scientists in the region to obtain specimens. And very rapidly, because this thing moved very quickly on the scale of only a week or so. We had to organize with some scientists in Ceiba in the Caribbean Netherlands and at the University of the Virgin Islands in the USVI. And basically, we came up with a very rapid sampling plan, which included grossly normal or healthy looking individuals at sites where the disease was, obviously the diseased individuals as well, and then reference sites, which are areas well away from the disease front. And the idea there is that we should be able to differentiate between what is actually causing this disease versus what is simply sort of taking over the urchins as they become sick, all right? This occurred at these two different locations, very rapid sampling, dissection, preservation. You can see Alwyn Hokema here is in Ceiba, who's basically taking a draw of the blood, the thalamic fluid of these animals, organized liquid nitrogen dry shippers to go down to those countries, and essentially they were returned to labs at Cornell, the Fish and Wildlife in Florida, University of South Florida, and the USGS in St. Petersburg, Florida. So we didn't know what we were looking at, okay? And so we had to basically come up with a sampling protocol that would allow us to highlight anywhere in these animals that this disease could happen, all right? And so this is my little South Park version of a sea urchin. I'm a terrible illustrator, I apologize for that. But what we did is we basically organized to have tissue samples that were amenable for molecular biology, for DNA and RNA, coming from the body wall and the spines and gonads, the reproductive tissues, their intestines. We also organized with veterinary histopathologists, a very, very important part of any disease investigation to basically have samples that they could analyze by histopathology, which is basically looking at thin sections that are stained and looking for abnormalities on a tissue level. We also did some collection for transmission electron microscopy, but at the same time, we are also interested in environmental conditions. And so we got some samples for tissue biochemistry, gross appearance, what they look like, water column chemistry, you name it, lots and lots of different types of samples that we obtained from these. And we're still working with our collaborators in the region to process some of the aspects of those samples. So the first question that we wanted to ask is there anything really obvious in the area that was going on, from a climate standpoint, ocean currents? Were there any weird sort of circling water masses called eddies that were moving into the area? And so for that, we actually turned to satellites imagery, because that's the sort of best way to get a handle on broad scale regional patterns of productivity. And so on the top left here, you'll see a little animation. Each one of these little yellow dots indicates where the disease happened. They turn red as the disease occurred. Unfortunately, most of the region was covered by clouds pretty sort of early on in that investigation. And so we couldn't really say for sure whether there were weird sort of blooms of algae that were occurring at the time. But there may have been, at least at the southern parts, some of these very, very high chlorophyll algal bloom conditions coming off of what's known as the North Brazil current down here in the form of an eddy. But it was largely inconclusive. But there seemed to be nothing really consistent between sites. There was some freshwater runoff that was going into sites at some areas that were affected, not at others. At some sites, some of the divers reported seeing this. This is a photo that Mariah Sevier took that basically shows what she thought were bristles from worms. Not too sure about that. It's probably some decaying plankton bloom that's shoaled from offshore, no idea. And also a lot of sargassum. But the point is that it was only restricted to 1 or 2 sites. It didn't seem to be a consistent pattern where there was weird stuff happening, was not necessarily lining up all the time with where this disease was occurring. And so we sort of turned our attention to other aspects, notably microbiology, to try and figure out if there's anything different. So what types of microbes could a disease like this actually be? As I mentioned before, you have a microbiome. Every animal has a microbiome, which comprises billions and billions of microorganisms, everything from bacteria and some of these things called archaea, which look broadly the same. They're actually evolutionarily a little bit sort of older, a little bit different. There could be a microbial eukaryote, which is basically something more like a fungi, a protozoan. Some of these nice little things on the left here that you can see, these are ciliates. And also it could be a virus as well. Okay, this down here on the bottom left is a TEM image of viruses. All of these together make up the animal microbiome. And going into a disease like this, you basically can't know which one is which one to target. Unfortunately, the methods that we use for analyzing and comparing between healthy and diseased specimens are very different between each one of these. And so you pretty much have to go at this with as broad sort of sweep of approaches as you possibly can. So this gets back to the question, what can we look for at signs of disease? And looking at molecular biology, we have 2 different forms of nucleic acids. We have DNA, which is basically like your storage molecule within cells. And then you have RNA, which is essentially what is being expressed as genes, gene expression. Some types of viruses actually have RNA as their core genome. So we have to look at 2 different types of nucleic acids, regardless of what we're doing. There's this thing called the central dogma in molecular biology. This is how all cells on the planet pretty much function. We have the storage molecule of DNA. It gets converted into RNA, and then that in turn gets translated into proteins and building blocks of cells. And so we have to sort of target our approaches that would give us the broadest approach, a broadest sort of brush of what might be responsible. And so for that, we turn to a few different things, which basically have really complicated names that I'll explain as we go along. But viral metagenomics is one, so that's looking at viruses, both DNA and RNA. Amplicon sequencing is another thing that is used to describe basically looking at the entire bacterial and archaeal community. And then transcriptomics is looking more at the host and basically the genes that it is expressing in response to a disease like this, okay? We don't do protein as much simply because technologically it's actually a lot more complicated. And generally speaking, you have to have some information about DNA and RNA to make sense of it. So start out by these 3 different approaches. And so The first approach that we did was using this thing called transcriptomics. This is where we take genes, RNA, which is in all the cells that are being expressed in the urchins, as well as their microbiome, their associated microbiome. So we start out with normal sea urchins here, abnormal or urchins that are dropping their spines. We extract messenger RNA, which is basically the gene transcript of all the genes. We then perform something called Illumina sequencing, which is this really, really amazing new technology that allows us to basically get a huge amount of DNA sequence or RNA sequences, and that gives us a great power to be able to say what's happening. And then we put it into this really, really old-looking computer on this graphic here. They never look like this. They're usually a lot more up to date. But we do computational analysis to tell us what those DNA sequences actually mean. All right. So the idea behind this approach is it will tell us what these animals are actually experiencing and what it might highlight some of the stressors that they might be going through at the time. And this is what it looks like. And, I'm going to explain what a bar chart looks like, because I know that perhaps some of the audience might not be familiar with this, but essentially what you're looking at here is we have grossly normal in the green, and we have abnormal or the diseased animals in the red. And each one of these different categories is a different type of gene transcript. And off to the right here, we have a couple that are involved more in structure or unknown genes. And what you can see is the magnitude of this one is obviously a lot higher than anywhere else that's involved in basically forming new cells. The ones that were all red here were mostly in the red and almost absent in the healthy animals. They are all involved in basically complementation or immune function. And basically what this is telling us is that there is some immune system sort of response to whatever's going on. And they have some enhanced maybe replication of tissues, but we don't really know. Okay, so they know something's weird, they're dying, but we can't say for sure based upon these transcriptomes exactly what they're responding to. Okay, so that brings us back to what the microbes are doing. All right, what types of microbes might be there? For this, we turn to a couple of different approaches, the first of which I'll describe as amplicon sequencing. So what this does is we have basically urchins. They have a microbiome associated with them. From that, we extract DNA. We perform something called the polymerase chain reaction, which is a way of amplifying a single sort of gene from a mixed community and then being able to work out the identity of that community based upon gene sequences. And again, we put that through Illumina sequencing in this 1980s computer. And essentially, it can tell us what different types of bacteria are there. And by comparing healthy versus diseased specimens, we can actually maybe identify what might be going on. What this told us when we did this approach is that, so at the top here, we have the unhealthy ones or abnormal ones. We have the ones that were normal at the sites that were affected. The reason that we separate those from reference sites is because sometimes you get animals that are infected by pathogens, but they're not quite showing disease signs yet. All right, so they might have the pathogen in their tissues, but they're still not quite sort of outwardly disease-ridden. And then we have the reference sites, which are the totally healthy ones. And what this told us is that there's really only two big differences between the diseased ones and the healthy ones. One of which is this. This is a Vibrio, okay? And in this case, this is a genus of Vibrio. There's about, I think, 18 different species within that one sort of part of this chart here. So nothing distinctive that was basically consistent between any of the urchins, just a generalized sort of response of Vibrios, which are basically weeds. They eat sort of decaying tissue, high nutrient environments, et cetera. But there was also this interesting one, all right? This is called Cystinophilium, which belongs to the Caddybacter group. Normally, this bacterium is an endosymbiont within a eukaryotic organism, okay? And this was kind of our first little ding, ding, ding moment, okay? So basically, we might be dealing with some sort of eukaryotic microorganism, something like a fungus or another type of protist, okay? And so that's basically what it looks like as far as the bacteria. We're currently refining this a little bit, but it definitely holds up across pretty much everywhere that we've been surveying. And then we look to viruses because I've got a long history of looking at viruses in aquatic habitats. That's what I did my PhD on. They're really hard to work with and to diagnose. So basically, what you have to do is you take the animal with the viruses potentially in it. You basically homogenize the tissue. You filter out everything that's larger than a virus. You then have to basically, excuse me, amplify the viruses somehow. In that case, we did so using a transcriptome kit, and then you sequence them. And we sort of tended to focus on the RNA viruses because those are the most important sort of animal pathogens. There are plenty of DNA viruses that are there that we pick up as well, and we would have been able to see it in the signal. And what we found was this, okay? And hopefully I'm not scaring you off with some phylogenetic trees here. All right, these are called trees. What it's showing you is that those things in red are most closely related to things that they're close to on the tree. Okay, so basically, sorry, my throat's going. So basically, what you're seeing here are a lot of different types of viruses that are related to things like insect viruses. From our history, we know that these viruses are generally not pathogenic. These are viruses that tend to be distributed pretty much everywhere, and we're not expecting them to be involved in the disease. And so we really haven't pursued this much further. But, so that sort of led us down a lane. So nothing bacterial, maybe an endosymbiont, no viruses. But we were interested in eukaryotic microbes based upon that early bacterial result. And so what we did is we reanalyzed the transcriptomes that we had, these expressed RNA libraries from the host. This time, instead of looking at the host genes, we looked at marker genes that would indicate what types of eukaryotic microbes were there based upon something called ribosomal RNA. And immediately what popped out is that there was this thing called Phylaster apodigitiformis, which is a type of ciliate in the diseased tissues. Maybe a little bit more in the diseased tissues than the others. There's some sort of ambiguity there because it's a little bit hard to explain. Basically, you can get things that look kind of like the ciliate, but they're actually not based upon different cutoffs in terms of DNA similarity. But we were really intrigued by that because it seemed to be in the sick ciliates. This particular bug, Phylaster apodigitiformis, is a known fish pathogen. It was basically retrieved from pufferfish eggs in China about 4 or 5 years ago. And basically, it was the only eukaryote that had any pattern of association with this particular condition. And so what's really cool is that immediately after we got this result, we started to talk with the veterinary histopathologists and cytologists, basically to see what they had found, because at this stage we were, about a month and a half in. We'd done 3 different independent lines of investigation. We weren't talking to each other so as not to bias the investigation for one particular pathogen. All 3 research groups came up with this exact same thing. Okay, this is Phylaster apodigitiformis. It looks like this. It's a big cell, all right, several tens of microns long. It's also pretty wide, and it's covered in these cilia, or these tiny little hairs that it beats to move around. Okay, this is one of the most successful groups of microbial eukaryotes on the planet in terms of their diversity as well as their distribution. You can find them pretty much everywhere. In terms of histopathology, what you're looking at on the right-hand side is a very, very thin section through a spine of a diseased animal. And all of these little things here, these little oval shapes, many, many, many thousands, if not millions of them, are all exactly the same thing. This is Phylaster apodigitiformis. We also did something called cytology, which is looking at the coelomic fluid and figuring out what types of things were around there. They were loaded with the ciliate as well. Okay, so we were like, this is very interesting. It was only in a few of the healthy animals from the affected site. Notably, it was not at any of the reference sites, the clean sites. Okay, so this is our first piece of evidence that this might actually be associated with this particular condition. We also did something that we wanted to see whether it was the same across all of the different sites in the Caribbean, this one particular type of ciliate. What you're looking at here is another phylogenetic tree, and the idea is on the sort of the horizontal axis, these things that are all together in this big branch here are identical, okay? And each one of these flags represents a different jurisdiction. As you can see, they range pretty much from Ceiba to Grenada, all right, right across the Caribbean, and they're all exactly the same ciliate, okay? So the same ciliate's there, it's distinctly associated with it, and we also had a couple of other pieces of evidence, like things in the transcriptome that were identical to to this particular pathogen as well. So this gave us good ammunition to try and understand what's going on. One thing that we also did is we took a very much wider range of samples, several tens and tens of samples, and we did something called quantitative PCR, which is a way of working out the abundance of a particular organism based upon PCR. And we did this on these reference sites. I know this is a big, scary figure. I'm really sorry, but basically all the bars on the left here are the reference sites. We have these normal and affected sites. So these are the healthy ones, which are cohabitating with these diseased ones. And then the ones in gray on the right, which are the most important ones, are all the abnormal. And what sticks out is that this Phylaster apodygitiformis is really in the diseased ones, okay? It's absent from the reference sites mostly. And again, there's a little sort of ambiguity there because of similarity based on nucleotides. And it's in a few of the healthy ones at the disease sites as well. So this numerically basically gave us an association between a microbe and the disease. But even if you find a microbe associated with the diseased animals exclusively, it might not be a pathogen. It might be some microbe that recruits from the environment that happens to take over hosts that are perhaps a little bit damaged or diseased or because of something else, okay? So in order to show that something is definitively associated with the condition, you need to satisfy what are known as Koch's postulates, a series of criteria that were put out by Robert Koch in the late 1800s, which beyond reasonable doubt can prove that a microbe is responsible. But a necessary first step is that you have to actually have this microbe, candidate microbe, in culture. And so we waited and we waited. Transporting cultures of microbes across international boundaries, particularly, thank you TSA at, where was it, Miami Airport for seizing everything that I had, but whatever. It's really hard. So we waited until this condition actually showed up in Florida. And it did one day in June, or sorry, late May in 2022 at one site. This is Key Largo here. This is Florida Bay and the Florida Reef Tract here. At this one site, it turned up. We had Fish and Wildlife ready to go with culture media. They were waiting for it. They took a diseased urchin. This is actually the diseased urchin called FWC2 from which it came. And we managed to get into culture on the first attempt. It's really easily cultivatable, this organism. We did dilution incubations to get it isolated. And then we now have a uniculture of this one particular cilia. We then happened to have access to a bunch of Diadema antillarum that were totally naive. They had never been exposed in the wild. These are from an aquarium system, the Florida Aquarium, rearing facility in Apollo Beach in Florida. And they very generously gave us 20 individuals. And what we did is we actually took those, we split them up into individual tanks. We took half of them, which we exposed to this one cultured ciliate of isolate of Phylaster apodygitiformis. We took another 10 and we basically filtered the culture. So there's bacteria and other things that might cause disease in the culture medium. And they basically, it's removed by filtering over a 5-micron filter. And we monitor them daily. This is a couple of our collaborators. This is Jim Evans, who's a postdoc at USGS. And this is Chris Kellogg, who is Jim's advisor. And we found that it actually killed them. 60% of the tested, so 6 out of the 10 individuals, they died. They had spine loss, and basically we harvested them and we looked for this particular cilia in their tissues, and it was going nuts in there. Okay, and so we were able also to take the cilia, the tissues at the end of the experiment from several of the diseased individuals and re-isolate the pathogen and re-identify it based upon genomics. And so this satisfies Koch's postulate. So we can definitely say for sure that the 2022 diadema mass mortality that we were observing was caused by this one particular cilia. So it's kind of a laborious and time-consuming, generally sort of way of proving that a pathogen is responsible for it. Normally, this process either never gets solved or it takes a super long time because you can't always culture everything. We did this in 5 weeks because we had access, right place, right time. We got lucky, okay? But it was a very quick investigation. So what can we say about it? So we can say that we have identified the causative agent of a major marine invertebrate mass mortality. This is also the first time any ciliate at all has been shown to result in any marine invertebrate disease. Okay, so normally they're fish parasites. In this case, it is definitively a invertebrate disease, an echinoderm. And we also, one of the big take homes was we established a really awesome collaborative network, international network of researchers from across the region to rapidly tackle future marine disease events. So we have the infrastructure in place to allow a response to any new diseases that might come along. And these things are absolute monsters. There's still a lot we don't really know about it. This is looking down the microscope just the other day. What you're going to see here is essentially, these are some free living ones. And what these things are consuming here, and there's a whole bunch of them around this big glob of brown, that's actually diadema tissue. This ciliate eats diadema tissue. It's called histophagy. It eats tissues. And we don't yet understand why it does so, because normally the ciliate eats bacteria. And so there has to have been something which caused a switch in its life cycle between something that eats bacteria and something that eats tissues. And we're still not sure exactly that. That's work that we have underway right now. Okay, so this leads to a couple of other questions that I'll just finish up with. Why now? Has something changed? What can we do about it? These are ongoing questions that we're working with various partners around the region on. We were very interested to see, can we actually detect the ciliate elsewhere in the environment? And so we performed 3 different field samplings in St. Thomas and St. John and Aruba. Went out with no expectation that we'd be able to see this ciliate elsewhere in the environment. It seemed relatively new in the urchins. It was not at the reference urchins at all. Swabbed just about everything from boat hulls to other species of urchins, corals, algae, fairly indiscriminately. I'm a microbiologist, bear in mind. I sort of go after what I know that I can identify, right? So, but we had a huge collection, about 500 total samples. And unexpectedly, drum roll, we did detect it. And it was really interesting because it is a coral-associated microbe, all right? We consistently found it in corals, and particularly one species of coral, the Siderastria siderea. This is a species of massive coral, which is throughout the entire region. It's generally considered a one that will grow in very harsh water quality. It occurs in marinas and harbors and ports and that kind of thing. And where we found it especially enriched is in the few specimens that we had this coral disease, all right? It means essentially that this might be a pathogen which was basically incubated or amplified, if you will, in coral disease. And it just so happens to have taken off and taken over in diadema populations. But we're still trying to understand that. That's the focus of a big NSF project that we just got knocked back on and we're hoping to get funding for in the future. Please, if you're a reviewer, give us money. All right. We've also done some experiments to see what different types of corals this phylaster actually will grow on. We've taken corals from various marinas through the Florida Keys, through the Florida Keys National Marine Sanctuary. We gave them sort of a choice as to where they would go. We added the ciliate to these different corals, and sure enough, they definitely grow on a few different specific corals, and they will completely ignore others. A big question is, how long has phylaster been in the Caribbean region? We can now say that it's been at least since 2019. We're able to see it in older coral specimens that we've been testing for the presence of this ciliate. And the big question is, where did phylaster come from? We don't know, and we may never know. Okay, it's really hard to track down biogeography of this thing. It may have been there for a really long time, but we don't have the specimens, for example. But we do know that it was on the US West Coast, particularly around San Diego and Los Angeles, during the 2013 to 2015 sea star wasting epidemic. So that was a big event that wiped out a huge number of sea stars on the West Coast that I was involved in. And sure enough, we can find this thing out there at the time. Probably not involved with the disease. That's a little spoiler alert, but the fact that it was there is really interesting because it means that it probably came from the Pacific to the Atlantic at some stage, or vice versa. So what's happening now in terms of disease? Unfortunately, it has jumped. All right. There was disease reported in the Mediterranean Sea around Turkey, Greece, Cyprus, and Israel, starting in summer of last year. There was also some reports of Madeira off of the coast of Africa a couple of months before that. Since that time, earlier this year, it spread into the Red Sea and particularly into the Gulf of Aqaba here. Our great colleagues in Eilat observed it, as well as in Jordan. And since that time, unfortunately, just in the last couple of months, it's made its way to Reunion Island, which is a small island near Mauritius off the coast of Madagascar. So this thing is moving really crazily across boundaries that it shouldn't be doing based upon natural history. Okay, to put it in perspective, this is what it looks like. It moved here. It was here in the middle of last year, started towards the middle of last year. It sort of jumped over here to the Madeira, the Eastern Mediterranean. Since that time, it's moved into the Gulf of Arabia. Okay, and we've confirmed that it is exactly the same parasite on a genomic level. And then it's down here in Reunion Island, and then just yesterday over here in Tanzania. All right. This doesn't move by currents. All right, it couldn't be moving on currents because this is against currents, and it's not consistent with the timeline. It has to be through some kind of vector. And I'm not going to say publicly what I think it is, but let's just say moving through the Suez Canal, there's not a lot that goes through there beyond what humans are doing. Okay, so we are currently trying to come up with sort of resolution on whether it's actually being moved around by people or not. So just to summarize, because I realize I'm out of time, we have rapidly identified the cause of diadema mass mortality across the Caribbean. It's this ciliate called Phylaster apodiditiformis. Appears to be a very highly prolific and virulent pathogen, can exist in environmental reservoirs, notably corals, and it can move very wide geographic distances, probably aided by humans. Little spoiler alert. What can we do about it? We're currently working on treatments for it, but the most important thing, in my opinion, is to stop it spreading. And that requires knowledge of how it's being moved around and involvement of management policy, which is unfortunately a little behind in terms of time. So with that, I would just like to thank some of the amazing cast of characters that I've had the opportunity to work with. So this is my lab at the top here. Brian is my graduate student. Brandon was working in the lab as a grad student last year, technician, a large number of undergraduates we work with at Cornell. Super proud to contribute to their training, as well as other sort of trainees in USF, USGS, and collaborators all across the region. There's too many people to put on one slide. But with that, I'll say I'll take any questions. Thank you.
Thank you so much, Ian, for that really exciting talk and really interesting work that you're doing. So thank you so much for sharing that with us. We have a few questions coming in the Q&A, and I have a few questions as well. So maybe I'll kick things off because I was really interested. I mean, your conclusion was to either have a treatment or to stop the spread, but I wonder, can you transplant back into the environment after these mass mortality events to try and recover the population? And thinking about your challenge experiment where you had 10 animals and 4 of them didn't show disease, are those animals more naturally resistant, and could they potentially be ones that you could transplant to recover the population?
Those are super, awesome questions. So the first question about transplanting or restocking efforts, there are efforts underway across the Caribbean to try and grow these things in aquaculture systems. So essentially, I know, for example, Alwyn Hokema, who's at Wageningen, who's based in Seba, has a big facility there trying to grow these things up. And the big question is, if you reintroduce them, will they get sick again, right? And that's kind of a question that we really kind of can't answer at this stage, because we know that the pathogen is probably still out there on coral surfaces. But that leads into the second question of, why are some seemingly resistant, all right? We've actually been looking at patterns of experiments that have been running to figure out challenge experiments that didn't work versus those that did work. There is an association between water quality, for sure. The question is how, and that's a really good question that I can't answer at this stage. We have an inkling that it basically involves the microbiome of the diadema itself and what's naturally there versus what gets disrupted under certain water physical chemical conditions. But that's something we're going to have to work on in the future.
Okay, some questions from our audience, and the the first 3 questions that came in are all pretty related to the spread of it. And the first one comes from Jonathan Pruitz, who asked, do you think it's associated with ballast water potentially fouling organisms on hulls? Is that?
Am I going to get into legal trouble for saying this? In short, ships seem to be a reasonable hypothesis of a vector. Whether that be ballast water, whether it be hull associations, which obviously these things get washed off as the boats move, but it could also be firefighting wells where you got a lot of stagnant water and these things tend to thrive. But honestly, we don't know at this stage. We don't even know whether this organism can actually survive under the conditions of, say, ballast water. That's something that's being investigated by some of our collaborators at the moment. But somehow it seems to be moving across geographic boundaries and against currents and things, which would suggest that it is being moved around.
And related to that, a question from Dylan Combs says, if hypothetically it's being spread by human activity, what are the recommendations and how to prevent its spread? And I'll follow on with the second question from Ian Gardner, is there a way to clean the bottom of ships as it moves around the world? So they're kind of connected a little bit. What can we do about it?
Well, technically speaking, and I don't know this for sure, I'm no expert in shipping at all, but My understanding is that ballast exchange is supposed to occur offshore, away from ports and away from coasts. And that might in some way sort of ameliorate this if perhaps there are boats which are not doing that. I realize that boats, when they are offloaded, have to take on ballast to maintain position in the water. And that might sort of move to another port. So a simple answer is change your ballast water, all right? Cleaning the hulls of ships, not as easy. I believe that there's a lot of biofouling that can happen. I don't know how frequently they're de-biofouled, but that would be one way to at least reduce the amount of material which they're bringing from port to port. In terms of firefighting wells, I believe that they already try and purge those every so often. So basically all ships have this sort of reservoir from which they draw water to fight fires on board the ship. And my understanding is they replenish that fairly frequently. So maybe that's not such a great mechanism for this spread. The other thing I will mention is one possibility is scuba divers moving from place to place. And particularly, I don't know if there are divers in the audience, when you are cleaning your dive gear and you have seawater in your BCD, okay, unless you clean that out really well, it might actually incubate in there. And then when you go diving somewhere else, it might come out. All right. So make sure you clean your dive gear really well. We have protocols in place where basically we take our stuff, we soak it in weak bleach solution, which will kill it for sure. We've seen that in culture. And we dry it out before we go somewhere else because we don't want to be responsible for it. So yeah, I think there are best practices out there, and hopefully more fundamental understanding of how this thing behaves in these kinds of industrial or human or built environment settings will help come up with those better practices.
Great, thank you. A question from Keith Wharton. Are there any ciliates that infect freshwater fish, such as zebrafish? And if so, have any susceptibility and resistance mechanisms been identified using model organism genetics?
I actually do not know the answer to that question at all. I've investigated them in some species of fish in marine habitats, but not so much in freshwater systems. What I can say about this organism, when it infects these things, one of the reasons that this is an interesting one is because it only affects Diadema and the family Diademidae, the urchins. Other urchins, things like collector urchins that are nearby and touching them, they're fine. And the reason for that is that we think, and we haven't really shown this from a temporal standpoint, all Diademidae have hollow spines. All other species of urchins do not. They're totally solid, okay? And it appears as though this organism colonizes the interior of those hollow spines, and then basically that is like a hypodermic needle into the tissues that underlie them, and then that's how it gets into the animal, and then essentially they die from that. So the actual process by which these ciliates consume the flesh of their Diadema seems to be fairly nonspecific because we find them everywhere in the animal, and it happens really quickly. So it's not like they're targeting one particular organ, but we don't have any model systems to play with. And yes, we have tried it against some of the model urchins as well to see if it's pathogenic, and it's not. It really is just these hollow spine diademas and their relatives.
Great, thank you. The next question comes from Dylan Combe again. Coral reef biologists debate as to whether the past diadem die-off or a decrease in the reef fish population is more to blame for the loss of coral reefs in the Caribbean. Is there any ongoing experiments using current day die-off as a case study on this topic?
If I understand the question correctly, so indeed, the Caribbean region has been pounded for over 800 years by all sorts of things like eutrophication, overfishing, dredging, that kind of thing. And so it's in pretty poor shape to begin with, even before the 1980s die-off. And so, we often talk about this, the keystone predator is basically causing the shift. It's probably multifactorial. It's probably one that contributed to the shift rather than being the exclusive reason for it. A big question that I always get is, was this ciliate back in the early 1980s? And the simple answer for that is we can detect things that are highly, highly similar to this in samples that were from regions around the same time. But unfortunately, no specimens actually were taken at the time, or at least exist today. So the answer is we'll probably never know. But it is interesting that we are starting to look at things like microbiome changes in samples before and after at the same sites to see if there's anything distinctive that might give us clues as to what might be happening back then. And yes, you can amplify microbiome material from 50-year-old museum specimens. It's pretty cool. Not sure if that answers the question, but
Okay, a question from Eric Ignatz. It appears that the ciliate has only spread to tropical warm waters. Have you done any studies to look at the pathogenesis of this organism at different temperatures? And do you think it poses any threat to more temperate habitats?
So we have looked at temperature and its growth in culture, and essentially it has an optima of exactly where it is right now. Not surprising, most microbes are very sort of highly tuned to their conditions in which you isolated them. It does better at slightly warmer temperatures, but too hot, like what happened in Florida Bay this summer. It wiped it out. Okay, we couldn't detect it at all in any urchin, corals, anything. Colder temperatures, it grows really slowly. Something like 12 degrees, it grows at about half the rate that it does at 28. And so there is also this problem where there's a correlation between productivity and temperature. So most of the places that urchins occur in cold water are also super, super rich, productive environments. And that actually is what we believe influences the virulence of the pathogen. And so we haven't been able to, every expectation we're not going to see this disease go through diademas that are from relatively productive conditions, because there's so much material for this ciliate to eat in terms of bacteria that it might never turn to sort of histophagy within the diademas. It's only pretty much in oligotrophic conditions. But that, again, is a hypothesis that we're trying to test at the moment.
All right, we're running out of time, but maybe just another question. And this is going to come back to you had mentioned that we don't yet know the cause of the sea star wasting disease, which has been really impactful in the Pacific. And can you maybe comment as to why it's been so challenging to identify the infectious agent in this particular case?
So there's a number of challenges with that investment. I was involved with that for 8 years, and including the initial 2013 outbreak. So first off, we never had access to any reference materials. So basically what happened was the disease started and immediately policy clamped down to say we couldn't take any specimens from pristine areas. And so that really made it difficult for us to have anything to compare to. All of our comparisons were healthy to diseased at the same site. And as we know, there's preclinical infections, so you couldn't really identify anything. We thought it was a virus. We figured out very quickly that the analysis that we'd done that led to that was incorrect. We spent a long time backtracking on that, but there's still a lot of people that say that it is a virus, and it's like, it's not a virus, okay? We also were, there's a few other issues with it as well, many different species, many different players, but the point is there's nothing distinctive about sea star wasting from a microbiological standpoint. It seems to be a very response to some sort of environmental change. We believe that it's actually organic carbon inputs from primary productivity that drives bacterial secondary production. That in turn leads to hypoxia on their surfaces, and things suffocating is never good for tissues, right? But the evidence for that, we don't have any direct evidence for that. So it is a long, circuitous investigation, and until this day, we haven't been able to figure it out. But we do know that it is a seasonal condition. It comes on in the fall, and that's about it.
Well, thank you again so much, Ian. I apologize to the members of our audience. We didn't get to all the questions tonight, but we've run out of time. But thank you, Ian, for so generously sharing your time with us and sharing your exciting research with us at the Science Hour. And thanks to everyone in the audience for joining us this evening. And I hope everyone will join us again for the next Science Hour, which is Thursday, January 18th, where we hear from Ryan Fellon, who is co-founder and executive director of Revive and Restore, who will discuss her work bringing cutting-edge biotechnology to conservation challenges. In the audience, you may also have received our 2023 annual appeal, and we hope that if you're inspired by our Science Hour talks and the work we're doing at GMGI, you'll contribute and support GMGI's mission. And thanks to all of you who have been supporters of GMGI. Please stay tuned, stay in touch. We love hearing from you, and we look forward to seeing you again, either at our research facility here on Gloucester Harbor or virtually at our next Science Hour. So thank you for joining us, and good night, everyone. Thanks, Ian.
You're welcome. Sorry I went a little over time.
No problem.